It's yes. Now it's 10:05.
Yes. Perfect.
Okay, so hi everybody. Welcome to our DevMech webinar. Today is my greatest pleasure to introduce Dr. Yusuke Toyama. Yusuke had a formal training in physics and engineering in Japan at the University of Hokkaido and Osaka, and then he got his PhD in engineering with a work on plasma and nuclear physics. Then he embraced biophysics and mechanobiology during his postdoctoral career at Duke University under the supervision of Prof. Glenn Edwards and Dan Keyhart. In 2010, he joined the Department of Biological Science at the National University of Singapore and got promoted as an associate professor and appointed Dean's Chair of the Faculty of Science in 2018. He also has an appointment as principal investigator at the Mechanobiology Institute of Singapore since 2010 and became deputy director of the same institute in 2019. So Andrea and I have been knowing Yusuke since our postdoc at the Mechanobiology Institute, and that the opportunity to publish great research together in prestigious journals, including Nature Cell Biology, Nature Communication, and so on. And our collaboration is still ongoing, as demonstrated by our latest paper that got accepted in Frontiers last week on the role of the endoplasmic reticulum in cadherin-mediated adhesion mechanotransduction. Anyway, today Yusuke will present his story and the latest findings of his group on the mechanical role of apoptosis in tissue morphogenesis and homeostasis. Thank you, Yusuke, for accepting our invitation.
Thank you very much. Thanks, Christina, for the introduction, and thanks, Andrea and Christina, for inviting me. It's my great pleasure, and I wish to go to Santiago, but I think I will keep that for the future. Okay, so now, today I want to talk about tissue dynamics. And basically, many scientists has been curious about how the collective cells behave. And this is the emergent property of the collective cells. And then this collective cell dynamics has been found in not only the development, but also the pathological conditions. So we have been studying on the mechanobiology of tissue dynamics. And basically, there are two key way to look at this dynamics. So the first one is that how those cell dynamics emerge based on the biochemical signalings. So this is represented by this arrow downside. And another important factor, that way of looking at the system, is that how the mechanical factors influence the biochemistry. So to understand fully that the tissue dynamics, as I already showed you, we need to understand those both arrows. So now I will come back to this cartoon a couple of times during the talk. Now, so in our lab that we have been working on the apoptosis in a tissue. So as you know, that the apoptosis is a mechanism to eliminate unnecessary aged or damaged cells. And we don't consider it too much, but each day, more than 10 billion cells undergoes apoptosis in our body. So certainly that if it's the apoptosis goes wrong, it will lead to the pathological conditions such as cancers. So not all the cells behaves in this way, but when the cell die in the tissue, the cells, unwanted cell, those dead cells will be kicked out from the epithelial cells. So this movie right here on the left show that how the dead cell, which is highlighted by red, is removed, pushed out from the neighboring cells to the apical regions. So this phenomena, we call this as a cell extrusion of apoptotic cell. And then this apoptotic cell extrusion can be found in our body too. And for example, in the tip of the intestine, the old cells, those epithelial cells are shedded or extruded, removed. And then that removal of the dead cell or the old cells help to turn over the epithelial cells. And then maybe you know that in this mammalian intestines, that the single layer of epithelial cells is renewed every 4 to 5 days. And then certainly if the extrusion goes wrong, that the renewal process also be compromised. So now, as you already see, that the cell extrusion process is in part, or mechanics play a major role. So we have been working on the mechanobiology of this apoptotic cell extrusion, especially in the context of how the cytoskeleton rearrange, or how the cell-cell junction rearrange, and how the dead cell and the neighboring cells, how the mechanical force are interacting, exchanging in between those two cells. So in this talk, what I'm going to do is I'm going to use one paper for the just introduction to explain that how the actin cytoskeleton, mainly the actin and the myosins, are changing in dead cell and the neighbors, and how the cell-cell junctions are rearranged, for especially the adherence junction, how the adherence junction are changing during the extrusion process. So that's what I'm going to explain as a part of the introduction. So later on, I'm going to explain that how the mechanical interaction between the dead cell and the neighboring cells are mechanically contribute to the tissue morphogenesis. And the third topic I'm going to talk about is our unpublished result, trying to understanding how that mechanical interaction influence the cell proliferation in the neighboring cells. And I will come back to that in the second half of this talk. Okay, so now the first topic, how the cells, the mechanical understanding of how the cell extruded. So for this topic, I need to introduce this developmental stage of Drosophila, which is so-called histoblast expansion. The picture you are looking at on the left is a confocal image of the surface of the pupa. And in this image, you see two different tissue types. One is so-called histoblast. So this is the future adult epithelial cells. And this histoblast is surrounded by the larval epithelial cells. So now during the metamorphosis from a larva to adult, these future adult tissues grow and expand. At the same time, those larval cells are no longer needed for the adult, so they disappear. They undergo apoptosis. So basically, there are two major biological processes happening here. One is in the cell proliferation. So this future adult tissue, as I already mentioned, that it's grow and expand. The growth is predominantly driven by the cell division. As you see that the the rounded shape cells here and there, that represent anaphase of the cell division. So the cell proliferation is going. And as I already mentioned, these larval cells, these huge larval cells undergo cell death. As you see that those cells are contracting and disappearing. And what that means is that this cell division and the growth and the cell apoptosis need to be well organized for this tissue replacement process. I forgot to mention that, interesting enough, that around 80 to 90% of the larval cells die at the interface in between those two tissues. So which suggests that there are some chemical interaction in between those two cells to trigger the apoptotic process, but I'm not going to explain that in this presentation. So instead, that what I want to focus is that the mechanical part of this tissue expansion. So here, this graph represents how the dead cell changing their apical section over time. So here, there is a wandering phase, and all of a sudden, the apical size start to constrict. As I already briefly mentioned, that the apical constriction is the hallmark of the apoptotic cell extrusion. So now the question is that when does this specific cell decided to die? So to address this question, we monitor the caspase activation. Maybe you already know that caspase is the very important signaling pathway, the process in the dying process, the apoptotic process. And then we basically monitor the caspase activity in live. So we use this FRET-based caspase sensor. Basically, there's a 2 fluorophore, and then those 2 fluorophores are connected by this small peptide. And among this peptide, there's a cleavage site. This peptide can be cleaved by the caspase. So once the caspase is activated, those peptides will be cleaved, and then those 2 fluorophore will be separated far enough so that the FRET signal will not be there. So basically, by looking at the FRET signal, you can measure that caspase activity. So in this image on the movie on the left, that the green represent the cell shape. Basically, this cell shape is highlighted by E-cadherin. And the yellow and the purple color represent the caspase activity. And if you look at these cells, those cells are greenish at the beginning, right here, and then it turned yellow. So which means that in the middle of this movie, those cells turn on the caspase activity. So now we can monitor the caspase activity and the cell shape or the area, the size of the cells simultaneously. So if we measure those two at the same time, what we can do is that we can monitor the cell shape as a function of time, which is highlighted by this orange, as I already showed you in the previous few slides. And we can also monitor how the caspase change over time. So in this graph shows that caspase start from low, and then eventually it goes higher caspase activity. So if you have those two graphs, what you can measure is that which comes first. And we conclude that in our system, in this system of histoblast expansion, that apical constrictions and extrusion of the dead cells are the consequence of the apoptosis, but not the cause of the apoptosis. So which means that those forced generation, the apical constriction and the forced generation is the consequence of the apoptosis. So now we know the caspase is activated in those cells. One of the consequence of the caspase activation is the cleavage of the adherens junction component, including E-cadherin. So in this movie, I'm showing that E-cadherin distribution, and this cell is going to die. So at this stage, that the cell-cell junction is very, very clear. Now, in the middle of this movie, you are going to see that this boundary getting fuzzy. So now again, that at this stage, that the cell boundary is very clear. And I hope that in the middle of this movie, right there, that boundary starting to get blur. So again, as I already mentioned, that caspase activated, caspase cleave the E-cadherin, and then it will reduce the intensity of E-cadherin from the cell-cell junction. In this movie, we're also looking at the distribution of cytoskeleton, in this case, myosin 2. So at this stage, you see that there is a clear cortical myosin at the cell-cell junction. And then in the middle of the movie, you are going to see that this single line is going to split in two lines. So now at the beginning, this myosin is a single line, and in the middle, right there, I hope that you see that this single line is split in two lines. So what we already know is that during this apoptotic process, there's a two actomyosin cable form. One is in the dying cell, and then the other is in the neighboring non-dying cells. And in the neighboring non-dying cells, there's actomyosin cable formed in the individual cells, and then those cables are linked through cell-cell junction. So the outer ring is a multicellular actomyosin cable. And the contraction of those actomyosin cables are essential for squeezing out this dead cell. I forgot to mention that those cells are squeezed out in the basal side of the tissue. So now the reason you see the two lines here is because at the beginning, the cell-cell junctions are tight. So those two cables cannot be, are very close to each other. So now in the middle, the cell-cell junctions are weakened. So cell-cell junction detach. So because of this physical detach, those two cables are separate physically. And then so that's why you see the open space in between those two lines. So which means that again, those two cells start to detach in the middle of this apoptotic process. So what the cell-cell junction detachment means. So in the tissue, there are pretension. So you can consider that the tissue as a balloon. And then which means that there is a pretension. And then if you poke and if you make the crack, the pretension will be released. As I already mentioned in the Drosophila case, that the cell-cell junction in between the dead cell and the neighboring non-dying cell will be detached. So because of this detachment, there are the formation of those two cells are being separated, meaning that it's considered as a crack formation. So which means that potentially that the pretension in the tissue could be released because of the cell-cell detachment. So to further address this question, whether how the neighboring cells, neighboring tissues change the mechanical tension in the course of the cell extrusion, we first monitor how the cell shape change during this process. So at this stage, this cell is a dead cell, and then here's the neighboring cells. Before the disengagement, we notice that the cell-cell boundaries is rather straight, which means that it's under tension. Right after the disengagement, meaning that the cell-cell junction detach, that the neighboring cells neighboring cell-cell junction tend to becomes wiggled, which means that the tension could be released at that point. Later on, that actomyosin cable form and then constrict. During that constriction, that the cell-cell junction straighten up again. So based on the morphology of the neighboring cells, or more specifically cell-cell junction in the neighboring cells, we hypothesize that that right after the disengagement, the tension in the neighboring tissue will be dropped, and then later on it will recover because of the actomyosin contraction. So to prove this, we further carry out the laser ablation experiment. And then basically, once we ablate the junctional cell-cell junction by UV laser, by measuring how fast those nodes move, recoil, that will suggest that what kind of tensions are there before the ablation. And which means that the faster the recoil velocity, higher the tension before the ablation. So what we have done is that we have done the laser ablation at the different stages, 3 different stages. And then we found that right after the disengagement, the recoil velocity dropped. And then during the constriction phase, that the junctional tension goes high again, or recovered. So the the morphology of the cell-cell junction in the neighboring tissues and this junctional tension variation both agree that in the middle of the apoptosis, when the cell-cell soon after cell-cell junction detach, that the neighboring tissue, the tension in the neighboring tissue dropped, and then later on it recover again. So here's a quick summary of what I have been discussing, is that during the course of the apoptosis, the caspase is activated. That caspase activation lead to the weakening of the cell-cell junction. And then that will lead to the cell-cell junction detachment, disengagement, and then that will lead to the tension reduction in the neighboring tissues. Later on, there's actomyosin cable form, and then they constrict. That constriction itself will recover the junctional tension later on, and then eventually those two neighboring cells form a de novo cell-cell junction. So we published this a few years ago, and then now there's two questions I want to address. So how this apoptotic process mechanically contribute to tissue dynamics during development? I already mentioned that the tensions are elevated in the neighboring tissue. So how that help the tissue dynamics during development? So that's the first question. The second question I'm going to address is that how the modulation of tension in the surrounding tissue further influence the fate of neighboring cells. So these are the two questions I'm going to address. So now, to address the first question, how this apoptotic process mechanically contribute to the tissue dynamics, I need to introduce other developmental stage of Drosophila. So in this case, so what you are looking at, a confocal image of the Drosophila embryo. And you are looking at the backside of the Drosophila embryo, and here, this eye opening, this tissue with eye opening shape is so-called aminoserosa cells. And during this so-called dorsal closure, what's happened is that those tissue, the aminoserosa tissues, are apically, the apical side of the cells are constricting. At the same time, some of the cells undergoes apoptosis. And eventually, this eye opening, all the cells will disappear, and then those epithelial cells in the neighbors sealed up, and eventually it will the embryo itself is covered by this epithelial cells. So as I already briefly mentioned, that some of those cells, the cells in the aminoserosa cells, undergoes apoptosis in the course of dorsal closure. What we already know at this point is that there's no cell proliferation during this dorsal closure, and only 10% of those cells undergoes apoptosis during the dorsal closure. So now, what I did was that I look at how the morphology changed during the course of cell extrusion. So this data shows that this is a time, and this is Z plane. So the 8 micron means 8 micron deep side of the epithelial cells. So what we found is that apical sections constrict, and then at the same time it's dive into the embryo, and then eventually it's bleb, or it's segmented as an apoptotic body. So this is basically the hallmark of the apoptotic process. So now if you look at the shape of the apical section of those cells, so the white represent a dead cell, and as dead cell constrict the apical surface, if you look at the neighboring cell, which is highlighted by red, it looks like as if that those red cells are being pulled or stretched in the direction of the dead cell. And in addition, those neighboring cells, which is not directly attached to the dead cell, these blue cells, are moving their location more closer to the dead cell. So those morphological changes suggest that potentially the mechanical stress in the neighboring cells increase. So I already told you that there's only 10% of the cells undergoes apoptosis during this dorsal closure. And then 10% could sound small number to you. However, those 10% of the cells are surrounded by the 6 to 7 cells. So which means that potentially the vast majority of the tissues are influenced by this small number of the apoptotic process. So our hypothesis is that if we inhibit or induce apoptosis in that tissue, potentially it will influence the dynamics of the dorsal closure. So what we did was that we induce the cell death in this tissue, or we inhibit the cell death in the tissue. And we compare the speed, the dynamics of this dorsal closure. So on the right that I'm plotting that the height of this tissue or the width of this tissue as a function of time. And what we found was that if there's more apoptosis in that tissue, the dorsal closure speed up and they close faster than the control. And if we block the apoptosis, it's the dorsal closure process slow down, but eventually it's do close. So basically there's a correlation in between the number of cell apoptotic process and the speed of the dorsal closure. So that will lead to the next hypothesis. Potentially the tension in that aminoserosa tissues are increased and it's correlate with the speed of the dorsal closure. So to address this question, we have done the laser ablation again. In this case, not the junctional ablation, but the tissue level ablation. And then we measure the recoil velocity, how fast this wound opens up. And then by measuring that force, that will tells you what kind of force are exerted in this tissue before the ablation. And then what we found was that in this graph that the y-axis represent that tension in the tissue, x-axis represent the number of apoptosis. And this graph clearly tells if there's a more apoptosis, the tissue's tension is higher. And if there's a less tension, the tissue, less number of apoptosis and the tension in the tissue goes down. So again, there's a clear correlation between the number of apoptosis and the tension in the tissue. So far, what I showed you is that there is a very clear correlation in between the number of the cell death and the velocity or the speed of the dorsal closure and the number of the apoptosis and the tension in the tissue. So based on those correlation, we conclude that apoptotic process can generate the force and the increase the tension, and then which will lead to speed up the number of the apoptotic process will control the tissue tension, and then tissue tension will control the velocity of the dorsal closure. So again, based on this correlation, we conclude that there's a new mechanical role of apoptosis. Previously, the apoptotic process has been considered as a passive process, meaning that just eliminating unwanted cell. But that from this study that we published more than 10 years ago, we proposed that the evolution basically trying to take advantage of every single sources of mechanical forces for the morphogenesis. And those apoptotic process convert to the mechanical force could be the one such forces. And then we call this as apoptotic force. So this is my study, what I have done during my postdoc. And then this was me. He looks more younger and it potentially could be energetic. So, and then now more than 10 years later, so now what is good about this time for more than 10 years is that this apoptotic force was not found only in the dorsal closure, but also the other process during the development. So for example, that this apoptotic process can pull the neighboring cells, and it can also contribute to bending the epithelial cells in 3D. So that was published a few years ago. And then we also found that the apoptotic process also contribute to the tissue expansion process, as I already explained to you in the histoblastic expansion. So now, hopefully, and then I think nowadays that there are some other paper speculating that this apoptotic process can be happening in the other morphological process or in the pathological condition too. So now that's the story about the how this apoptotic process mechanically contribute to the tissue dynamics, especially during development. So now I'm planning to switch a gear. I'm going to switch a gear to address the second question, how the modulation of tension in the neighboring tissue influence the biochemical nature in the neighboring cells, and which lead to influence the fate of the neighboring cells. So now this is the story about the compensatory proliferation, which is the relationship in between the apoptotic process and the cell division. So when the cell die in a tissue, the number of the cells reduce by 1. And to compensate the number of the cells, the neighboring cells need to divide. So this is the concept of the compensatory cell proliferation. And then this concept was shown more than 40 years ago. However, even at this point, we still don't know why only a very small number of the neighboring cells divide in response to the apoptotic process. So if all the neighboring cells divide, there's too many cells, so which means overgrowth, and which can be considered. That's what we can see in the context of cancer, for example. So again, the question we are trying to address is why only the small number of cells in the neighboring cells divide. How those cells are being chosen to divide. So that's the question we want to address. So to address this question, we have been using the MDCK cells, which is dog kidney cells. So now we are moving away from the fly story, and then we are now talking, I'm now talking about the mammalian cells. So a few years ago, we established the system in the lab to induce the apoptosis in the given cells. So we shed the UV light to the nucleus gently, and we cause the DNA damage. And if the DNA damage is severe enough, those cells undergoes apoptosis. So now we can induce the cell death in the given cells at the given time. So by taking advantage of this system, we are also interested in how the mechanical force are changed in the neighboring cells, neighboring tissue. So to address this question, so to measure the mechanical status of the neighboring cells, we have been using this attraction force microscopy. So basically, we prepare the gel, and then we embed the fluorescence beads in it. And then we culture the cells on top of it. And if the cells exerted the force to the substrate deform. That the deformation of the substrate can be measured by tracking the movement of the beads. And then now which means that by looking at by tracking the beads movement, we can deduce what kind of force the cells are exerted. The cells exert the force to the substrate. So the cells, the movie on the left, I'm showing you only the beads. However, in the reality that we culture the tissue on the top, but I'm only showing you the beads. So what we did was that we induced a cell death in the middle. And what we found was this beautiful wave. And this yellow circle represents the size of the single epithelial cells. So, which means that this wave propagation or the force propagation is not only at the very vicinity of the dead cell, but the 2 to 3 cells layer away from the dead cell. So now we have a capability, we have tools to measure how the force is propagating in the tissue. So now to cut the long story short, we conclude that this beads movement is not due to the cell migration. So we first thought that the neighboring cells crawl toward the dead cell. And if the cell crawl, that the cells push the beads backward to move forward. So we thought that this outward movement of the beads is originated by the cell crawling by the neighbors. However, we conclude that that's not the case. In fact, that when we inhibit the cell crawling by treating the tissue by this drug, NSC 23766, which is the RAC inhibitor, which compromised the cell migration. And in those circumstances, this wave enlarged instead of diminishing. So based on that, in part, based on this result, we conclude that that's not the case. The cell migration is not the cause of this beads movement. Instead, that we conclude that this is due to the release of tissue pretension. As I already explained to you, that epithelial cells are basically under tension, and then if you make the crack, that tension will be released. And then, as I already explained to you a few minutes ago, when I explained the histoblastic expansion story, that the cell-cell junction is weakened, and the cell-cell junctions start being detached, and then as a consequence, the pretension in the neighboring tissue would be released. So similar story in MDCK cells, and we monitor that a few years ago that we report that the cell-cell junction, especially in this case, E-cadherin, is reduced, the intensity is reduced in the course of apoptosis. So here, this is a dead cell, this is a neighboring cells, and then as you see that E-cadherins are, the level is reducing in the course of the cell extrusion. So meaning that the cell-cell junctions are going to be weakened during this process. So now on the image on the right shows that the tissue dynamics during the cell extrusion. So we induce a cell death in the middle. And I hope that you see that the neighboring tissues are moving outward. So it looks like as if the tissues are relaxing. So if you cannot see clearly from this movie, so here, this is analysis of the PIV analysis of how the tissues are moving. And then the old arrow pointing outward, meaning that the tissues are relaxing or moving outward, suggesting that the tissues are relaxing and then moving outward. So let me quickly summarize that what I have been explained to you. So in the tissue, there's a pretension. When the cell dies, the cell-cell junctions are weakened, and then the cell-cell junction will be detached. So because of that detachment, the pretension will be released, and then as a consequence, that the tension release will be propagated to the neighboring tissue. So that wave propagation is very, very interesting, also from the physics perspective. So we have been collaborating with 3 physicists, Yutin, Tetsuya, and then Jack Pro, to come up with a theoretical model for this wave propagation. But I'm not going to explain that in detail today. So now, what I haven't told you so far is that while the tissues are relaxing and they're moving outward, there are actomyosin cable formed in the neighboring cells, and then that cable will be constrict. And then that constriction will help to push the dead cell upward. So in this case, the mammalian cells, the dead cells tend to extruded in the apical section, apical side of the cells. So which means that relaxation of the tissue in general and the contraction of the actomyosin cable in the middle, the combination of those two will lead to the stretch of the neighboring cells. And in fact, that we monitor that how the neighboring cells change their shape. And here the blue represent the dead cell, the yellow represent the neighboring cells. And then we conclude that the neighboring cells change the strain, or in other words, that those neighboring cells are being stretched more than the cells far away from the dead cell. So the conclusion is that neighboring cells are being stretched. So now the question is whether this stretching of the neighboring cells, how possibly that stretching will change the biochemical nature in the neighboring cells. So that's a hypothesis. So to test this hypothesis, what we have done is that we monitor a couple of the localization of a couple of proteins in the neighboring cells. So in this case, that on top of the traction force microscopy, we monitor the localization of some of the proteins. So in this case, it's a YAP, which is the YAP associated protein, and the YAP is known to change the localization from the cytoplasm to the nucleus once the cells feel the mechanical force. So what we are trying to monitor is that when we induce a cell death, what's happened to the localization of the YAP in the neighboring tissue. So now we induce a cell death in this specific cells. And what we found was that a very small number of the cells show the nuclear localization of YAP. For example, in this experiment, these specific cells show the nuclear translocation of YAP, end up with a more brighter signal in the nucleus. So what we know is that once the YAP go into the nucleus, one of the consequences of this YAP nuclear translocation is the cell division. So what we did was that we monitor the cell cycle change after apoptosis. So in this experiment, we first synchronize the cell cycle, and then we use the Fuchi, which is the cell cycle marker. If the nucleus is red, it represents the cells are in G1 phase. And then once it's turned the color from yellow to green, which show the progression of the cell cycle from G1 to S to G2 and M phase, and eventually they divide. So again, at the beginning that we synchronize the cell cycle all to G1 phase, and then we lift the synchronization. And then later on, we induce a cell death in the middle. So this white blob in the middle represents a dead cell. So now our question is that what's happened to the color of the neighboring cells upon this apoptotic process? And what we found was only a very small number of the cells change the color from red to green, and eventually they divide. So this finding, which this data represent, again, only the very small number of the neighboring cells changing the cell cycle and then divide. So this is consistent with our finding from our YAP nuclear translocation data. So now what I show you so far is that only a small number of the cells in the neighboring cells upon apoptosis show the nuclear translocation YAP and the cell cycle progression. So now the question, now the next question is that where the inhomogeneity of cell divisions comes from. Why only the small number of cells are chosen to be divided? And we found two factors. And the first factor is that the pretension release is inhomogeneous. So as you see in this movie, although that it looks like a very beautiful wave, but if you quantify them by PIV, we start to realize that in some region that the beads movement is more in compared to the others. And then we start to realize that the cell division happen more at this region where the beads move, this movement is larger. So we start to see that correlation in between the beads movement and the cell division. So this is the first factor of the inhomogeneity. The next factor is the nuclear size. So here, we measure the nuclear size in the neighbors right before the apoptosis. And then what we found was that when the cells have a larger nucleus, those cells tend to progress the cell cycle from G1 to S2. And what we found was that those cells in the neighbors, which show the cell cycle progression, has the larger nucleus in compared to the others, which didn't show the cell cycle progression. And on average, the cells show the cell cycle progression have around 50% larger than the other cells. But we know that the nuclear size also enlarged in the course of cell division. So what we did was that we monitor that how the nuclear size change over time course of the cell division individually. And then we found that on average, that the nuclear size increased only 10% in the course of the cell division. The cell division means that before the M phase. And this 10% increment cannot account for the 50% difference in between the cell cycle progression group versus the non-cell cycle progression group, as I showed you in the previous slide. Moreover, we also monitor that in the snapshot, how the distribution of the nuclear size looks like. And then we found, so this graph shows that the y-axis is frequency and the x-axis is the nuclear size. And then we found that it's very diverse. And then sometimes that some cells have 3 times larger than the other cells. So which means that the nuclear size have intrinsic inhomogeneity. So we conclude that this size difference comes from the intrinsic inhomogeneity, which can be found in the tissue. So now the question is why the large nucleus tend to change the cell cycle. So that's the next question. So what we found was that if the nuclear size is large, there's a more nuclear pore complex. And then as you see in this graph, that a larger nucleus have a more nuclear pore complex. And then what we already know from the other study was that the nuclear pore complex is the pore which intake the YAP molecule from cytoplasm to the nucleus. So which suggests that the larger nuclear size have more capability to bring in the YAP molecule and then the other molecules from the cytoplasm to the nucleus. So now, so far I told you that there's a two factor. One is the nuclear size, and then the other is the B's displacement. So those two are the one which is correlated with cell cycle progression and the YAP nuclear translocation. So the question is whether those two factors are both required or only the one factor is enough to progress, to change the cell cycle. So in this graph, we plot the y-axis the nuclear size, x-axis the B's displacement, and the red dot represent the cells which show the YAP nuclear translocation. Blue represent that the YAP is still in the cytoplasm. And then what we found was that this red cluster was found only in the region where the cells have a larger nucleus and they experience large B's displacement. And in the other case, for example, even the cells experience a large displacement, B's displacement, but if the nuclear size is small, they would not show the nuclear translocation of YAP. And then the other case is if even the cells have a larger nucleus, if the B's displacement is small, they also don't show the YAP nuclear translocation. So which suggests that those B's displacement and the nuclear area are both required for the cells to change the cell cycle. So in this graph on the x-axis, I only show the B's displacement, but I already show you that the B's displacement is also related to the strain, how the cells being stretched. So in this graph, the x-axis represent the strain, and the y-axis represent the cell area. And the red, again, it's cluster in the region where the cells experience a large strain and large nuclear area. So again, those two data shows that those two factors, that the large nuclear area and then large mechanical force contribution, meaning that the strains are important for changing the cell cycle progression. So now what we also know is that the apoptotic cell could secrete the mitogenic signal, for example, JNK signaling. So how those mitogenic signal would influence, and then we already know that those chemical signaling also contribute to the cell cycle change and the cell division in the neighboring cells. So how about this? How about the role of the mitogenic signals? So this is, we already know that this signaling process is very complicated. So it's very hard to address. So what we did was that instead of directly address this question, we tried to indirectly address this question. So the strategy we adopted was our aim is to modulate the mechanical force propagation or the strain. So we are trying to modulate the mechanics but not the chemical aspect. So to do this, what we did was that we take advantage of the micro contact printing technology. So we make the pattern of the ECM so that the cell can adhere only in this region. So we can make the small tissue. Our intention is to reduce the pretension with a small size so that there is no more wave propagation. So what we did was we changed the size of this mini tissue, and we make it large to small. And then what we realized is that when the size of the tissue is big, for example, 500 micron diameter, there is still a wave propagation. But once it's reached 100 micron, and then those wave propagation diminish. So we adopt the cell mini tissue size of 100 micron, which means, as I already mentioned, there is no wave propagation, and then culture this tissue, and then we induce a cell death in the middle. And then what we found was that in this case, in the case of mini tissue, even there's a large nucleus in the neighbors, we don't see the wave propagation. And then in those circumstances, we don't see the YAP nuclear translocation or the cell cycle progression, meaning that we don't see the cell division in the neighbors. So which is in contrast to the more larger tissue, which in this case, that if there's a larger nucleus and if there's a larger mechanical stimulation, and then that will lead to the cell division, as I already explained to you previously. So now, one of the beauty of this experiment is that the chemical interaction in between the dead cell and the neighboring cells could be very, very similar in between this mini tissue and the larger tissue. So the major difference is whether there's a mechanical stimulation in the neighboring tissues, or there's whether there's a mechanical stimulation or there's no mechanical stimulation. So based on this experiment, this line of experiment, we conclude that the reason why only the neighboring cells, only the small number of the neighboring cells divide in response to the mechanical force is the following. So there's two major factors. One is that the neighboring cells is going to experience mechanical stimulation. And then if the neighboring cells have a larger nucleus, and then if the cells with a larger nucleus experience the large mechanical stimulation, if those two happen together, that's the scenario where the YAP will go into the nucleus and eventually they divide. So now we hope that I convince you that we found that the additional mechanism of the compensatory proliferation, especially this mechanism could explain, again, why only the small number of the neighboring cells divide, and then why those small number of cells are chosen to be divided. So I hope that now, I hope that you have some sense that the apoptotic process in the tissue is not just a passive, boring process, but many of the mechanics are involved. Okay, so let me conclude, let me finish here. And of course, that this study, line of study, has not been done by myself. And many of the previous lab members and current lab members have been contributing to this study. And we have been fortunate to have many good collaborators along to study this process, from fly study to the mammalian cell studies. We also want to thank the fantastic facility here from Wet Lab 4, Microscopy Computational Core, and the Nano and Microfabrication Core. I also want to thank the Drosophila community, including the range of the stock centers and the funding agencies. Thank you very much, and I'm happy to take your question if you may have. Thank you very much.
Thank you, Yusuke. Fantastic. Great presentation. I love, can you hear me?
Yes.
I especially love the last part where you're clearly showing that you do require a mechanical force in order to have the transmission of a chemical signal or a biochemical signal, if at all. So I have a first question, while the other warm up, which is related to this last part. So do you think that the cell with the large nuclei is also under stronger preload together with the cell that then undergoes apoptosis? So I'm trying to understand what you think in terms of causation and correlation.
Right. So your question is whether those cells with a larger nucleus, whether those cells already have a larger pretension.
Correct.
And how that linked to the pretension release, and then how that linked to the cell cycle change.
Right.
So that's I think that's a very good question. And then my simple answer is we don't know yet. So we don't know what's the pre-tension state before the apoptosis, and which is hard to address. And then I think in part that's also related to the size of the cell, not from only the nucleus. And if you look at, and then most likely that those cells with a larger nucleus may have a larger cell size.
Mm-hmm.
And potentially what that means is that there could be larger, the reason why those cells are large is potentially because it could be larger pretension, or it's basically the cells are simply big.
Right.
So we cannot narrow down which conditions are those large nucleus. But potential, yeah, so I think, what we know is that what's happened after the extrusion, and then, but we don't know that what's happened before.
The condition before, yeah.
Yeah.
Okay, thank you. So there is a question from Miguel.
Hi, very nice presentation.
Thank you.
So I was going to ask something very similar to what you were just saying, because you found that this induction of apoptosis was meeting at this large nucleus and large strain. And I wonder what is really generating the inhomogeneity in the strains within the tissue. Yeah. I mean, whether you have done kind of correlation. I was going to ask about, I mean, cell size or cell shape, because, yeah, and whether, as you have just said, whether really the size of the nucleus and the size of the cell is really a connection. Have you done any correlation between some parameters of cell size or shape with the strain?
Right. So we have not looked into that carefully yet, but I think as I think both of you asked, so I think the simple question is where this inhomogeneity of wave propagation comes from. And then I think to address that, potentially if we monitor, if we track down that how the cell size and then how the shape of the cells, if we can track this down, potentially we can start to see the correlation in between the wave, the inhomogeneity in wave, and inhomogeneity in the nature of cells, meaning that the shape and the nuclear site and so on. I think that will be the next step for us to understand.
Have you done some like local manipulation of strain to see whether you can induce apoptosis?
I think that will be cool. I think that will be cool. So one thing we have been doing is not directly addressing the local manipulation, but what we are trying to do is that we are trying to make So what I show you is this mini tissue is like a circle. What we are doing now is that we are making the tissue in the non-circle, meaning that in the ellipse, but a very long ellipse. So one side is 1 millimeter long, and then the other shorter axis is a few tens of micron. So our intention is to change the pre-tension in one direction to the others, and then hopefully to see that the inhomogeneity of the wave propagation could be enhanced in those circumstances. So instead of manipulating the small region, we are trying to make the manipulating the pretension in the larger scale, and then how that link to the shape of the wave.
Yeah, and sorry, the last question. So it's a more general question. What makes a cell to decide to undergo extrusion to the apical or basolateral side?
Yes, that's a fantastic question. So, so let me explain, let me address first in this way. So in general, in mammalian cells, the cells are extruded in the apical section, apical way, in general. And in Drosophila, those cells are extruded basally. And your question is, what determined this difference in the cell extrusion? And basically, that's what I want to know. And we still don't have a clear answer yet. I have some hypothesis, but I think it's just a hypothesis. And then I think we wish to address that question. But I think in short, we don't know.
Thank you.
Okay, thank you, Miguel. Carreta?
Okay, thank you. Yes, I'd also like to say, just like the two people before me, that was a wonderful talk.
Thank you.
Beautiful images, very clear, and it does show mechanics is behind most things in biology. So I'd like to go back to your days as a postdoc and to your extrusion, your dorsal closure model. You showed that there's 10% apoptosis, and that is then helping the cells actually be extruded. But when you actually inhibited apoptosis, you still got extrusion. So is that saying that apoptosis is not necessary? It's just involved in the kinetics? So that's sort of my first question. My second question is, do you think there's only 10% apoptosis if it is involved? Because more than 10% apoptosis would then create too big a space, too big a hole. If we think that maybe the cells are disappearing and the other cells that have to come in to take its place are pulled in, if there's more than 10%, it would create maybe a greater space than the cells could migrate or be pulled in. And that's why there's only 10%.
Yes. So let me address the second question first. So now, in the wild type, it's only 10%. And then if we induce the cell death very severely, and then the tissue integrity will be ripped off, and then all of a sudden the gut and everything will comes out. Basically, the cells die. So what I did was that I need to gently increase the number of the cell death. So I think I increased by a factor of 2, so meaning only double. I think that's my recollection. And in those cases, the tissue integrity is still there. And then in addition, that it's succeed to increase the tension and it speed up the extrusion, the dorsal closure process. So meaning that the number, if the number is way higher than 10%, as I already briefly mentioned, that the tissue integrity cannot be held. So that's the second question. And then the first question is about that when we inhibit the cell death. So I think maybe I explained poorly during my presentation that when we inhibit the apoptosis, that there is no extrusion during the dorsal closure. But after the dorsal closure, all the tissues are kind of embedded inside of the embryo. Is that answer your first question?
Perfectly, perfectly. I have another question just to throw out there, talking about the question that Miguel Concha just asked about maybe what is the actual initial signal for apoptosis, and you were showing that maybe when there is division afterwards, it's the bigger nucleus. Do you think there's any possibility that the cell with the bigger nucleus is the one sending the signal to the other cells to go under apoptosis, and then that is that cell then is the one that divides? I know you sort of disproved that slightly, saying you need the mechanical signal as well, but still, could there be, going back to the idea of cause and effect, and maybe there's a certain specific cell with certain specific tension that sends the signal and decides before anything happens what the cell is that's going to divide?
Right. So let's see. So one part of the question is whether cell division could induce a cell death in the neighbors.
No, whether the cell that's destined to divide is the one that initiates the cell death.
Right. So the apoptotic cell we examine, we don't necessarily see a very strong correlation of the cell division prior to the cell death in the vicinity. So I cannot rule out that possibility. However, based on the data in our hand, we don't see that correlation often. So, and so we don't think, so at least that in our data, we don't think that cell division could influence, could trigger the apoptosis. So now the question is how the apoptosis process trigger in the first place. I think that could be another question. And in our case, that we have done two studies. One is we induce a cell death by laser. So that's one. The other is that we just let them go, let them grow, and then time to time that the cell die spontaneously. So my answer that we don't see the cell division prior to the cell death is for the second one, which is a spontaneous cell division, spontaneous apoptotic process. And I don't recall your...
No, that was it. That's it. Yeah.
Okay.
Okay, thank you.
Yeah, I would also have some question, but maybe I would like to involve the students. If any of them have any question, would be great.
Be brave.
Otherwise, I can ask you in the meantime. So during the dorsal closure, there is no active process from the epithelial cells in the closure? There is no involvement there? Yeah.
Right. So let's see. In the dorsal closure, so the cells in the middle, they die. That's one. Yeah. And then they all... Say it again?
And they pull down, basically, right?
Yes, it's constrict. Yeah. The apical side constrict, and then some of the cell die. And then basically this constriction will pull the neighboring tissue to help the closure. So there's no basically pushing force from the epithelial cells.
Okay, there is no. Yeah. And then the other thing is exactly in this schematic that we see here, where there is the mechanical stimulation. I was wondering, because, well, obviously the obvious question was the one Andrea made about the cell with the large nucleus. I was wondering if you ever looked at the reorganization of the actin cytoskeleton at the borders between the apoptotic cells and the large nucleus respect to the others instead, so to the other neighboring, if there is a different reorganization of cadherin and actin cytoskeleton.
Yeah, I think that's a fantastic question, and then I think that's the one that we should look at the next phase. I think what we described here is still a very descriptive explanation, and then so I think we need to look into more detail, and including cytoskeleton of and then the cell junction.
Mm-hmm. Yeah.
Great. Are there any more questions for our guest? If not, I do thank you, Yusuke, for taking time in Singapore late in the night to give us this fantastic talk.
Yeah,
Thanks a lot.
great.
So I stopped the recording.
Thank you very much.